So good evening and welcome to the latest installment of the Torrey Botanical Society Torrey Lecture Series. My name is Catherine Mercier. I'm the Society Programming Chair. Before we begin, I'd like to give a little bit of background about the Society. So the Torrey Botanical Society was founded in 1867, and it's an organization for anyone interested in plant life, from professional and amateur botanists to students and anyone who simply enjoys nature. The objectives of the Society are to promote an interest in botany and to collect and disseminate information on all phases of plant science. And to achieve these goals, we have public lectures such as these. We do field trips. I'll be highlighting 2 upcoming field trips in just a minute. We also have the Journal of the Torrey Botanical Society. We fund graduate student research and education. Our grants for grad students and undergrads, I think, will be opening this month, so keep an eye out for that if you're a student. We also sponsor symposia and regional conferences. The best way to stay up to date with everything that the Society is doing is to become a member, which you can find out more about on our website, torreybotanical.org. And you can also follow us on social media. We are at Torrey Botanical almost everywhere. So we're at Torrey Botanical on Twitter, Facebook, Instagram, Blue Sky, and then on YouTube, where we post recordings of the lectures, we're at Torrey Botanical Society. Somebody must have had at Torrey Botanical. So to give you a little bit of, to highlight some upcoming talks. Oh, there we go. Some upcoming field trips. So on the 19th, we have a plant walk at Fort Washington Park, right around the corner from me. I think it starts at 10 a.m. And then the following Saturday on the 26th, we have lichens and bryophytes of Clay Pit Pond State Park in Staten Island. So check out those. The registration is on Eventbrite, but you can find the Eventbrite links on the website, torreybotanical.org. And I also want to highlight the special issue coming up, Botany and Ecology of New York State. The deadline for submission is at the end of the month, October 31st. If you have a study that you think could fit into this issue, submit it or reach out to our editor-in-chief, Dr. Carolyn Copenhever, whose information is also on the website, torreybotanical.org. I think that's all I have to highlight. In summary, visit torreybotanical.org. Before we begin the talk, I have a few notes about Zoom and Zoom etiquette. Please keep yourself muted during the talk. You're welcome to have your video on or off. You're welcome to enter questions in the chat as we go, but we will have a Q&A portion of the night at the end or as the speaker decides. If you would like to share this talk with someone else or rewatch it later, it will be recorded and available on the YouTube page. So I will stop sharing and introduce our speaker. So tonight's talk will be presented by Dr. Brad Oberle, who is an Associate Curator at the New York Botanical Garden. He earned his PhD from Washington State University in St. Louis, and before joining the New York Botanical Garden was a professor of botany and environmental studies at the New College of Florida. His work leverages the New York Botanical Garden's collections to address global problems of climate change and biodiversity loss through techniques from ecology, evolution, and statistics, and especially is interested in working in forests. And with that, I will hand it over to Dr. Brad Oberle to talk about Rooted in Resilience, Below-Ground Variation in the World's Premier Urban Research Forest.
Awesome. Thank you so much, Katrin, for the invitation and the introduction. It is a real honor to be with you here, speaking to the Torrey Botanical Society. I will share a quick anecdote right off the bat. I love the symbol. And actually, when I was living in Florida, right after our wedding, I would call it our honeymoon, but my wife would dispute that. We went to Torrey State Park and paid a kind of botanical pilgrimage to the amazing plants that were there. And that was a really special experience from my time botanizing in Florida, and something that clearly connects to the work that I'm doing here in New York at the New York Botanical Garden. I'll be talking about that work a lot more as part of my presentation. So, as Katrin said, the title of this talk is Rooted in Resilience, Below-Ground Variation in the World's Premier Urban Research Forest. So I'm going to talk a little bit about myself, a little bit about the work that brought me to the New York Botanical Garden, and some of the work that's been going on in the forest that you see in this picture at the New York Botanical Garden since its founding in the late 19th century. And specifically, I'll be focusing on a project that I've been doing with the great people in this picture. But this is a kind of project that has involved a lot of people over a very long period of time, and I hope to acknowledge them as I go along. So to begin with me and my research program at NYBG, I'm really interested in forest ecology and climate change research. And I came to this topic from a couple of different perspectives. The original one during my PhD was very much botanically based, and it was very much focused on plant biodiversity. The plant that you can see here, illustrated by a type specimen in our herbarium at NYBG, is French's shooting star, my absolute favorite plant on the planet. It's a really funky thing that actually grows only in the state of Missouri where I grew up, in a single cliff that was close to our family farm. I never saw it there, but just a funny little connection. This species is very vulnerable to the effects of climate change. That was one of the outcomes of the research project that I did when I was a graduate student. And unfortunately, no one loves those plants more than I do, and it was really hard to motivate people to act. So in my postdoc, I started to take a different perspective and thinking not just about how climate change impacts plants, but actually how plants affect climate change through their role in the global carbon cycle. And specifically what I was looking at is how wood decomposes and thinking about how variation and how wood is constructed, vessel anatomy, chemistry, physical properties influence the assembly of microbial communities, especially fungi, and then how they take the wood apart. So this was a really nice and complementary perspective to the role of plants in the global carbon cycle and in climate change. And it was really basic science research. That all became much more applied during my last gig at New College of Florida. As Katrin mentioned, I was a professor there for about 8 years. And before I move forward, I just want to take a quick moment and acknowledge people down there are having a really tough night. And it's very much climate change related. And I actually feel pretty good in giving this talk, not just to sort of speak to that challenging situation, but because like this is a picture of me trying to make a difference. I was here with some of the students at New College demonstrating the carbon cycle to a group of community scientists who were looking at coastal restoration areas where we were measuring carbon sequestration. And it's carbon in those coastal restoration areas, which I sincerely hope are protecting friends who live in that community right now. So I was doing all kinds of stuff in the community. I was the chair of the Tree Advisory Council. I helped establish a memorandum of agreement with a really great little botanical garden there, Marie Selby Botanical Garden. And of course, I got funding from a whole bunch of different places. So as you can probably figure out in my research background, the real themes are plants and climate change, and really trying to mobilize communities to make a difference. And so the question that really drives my work is how botany can enhance the resilience of diverse communities, communities of people, communities of plants, and the interactions between them. In this picture that looks out over the Thain Family Forest at the New York Botanical Garden, south towards the skyline of Manhattan, I think does a really good job of illustrating the sort of intimate connections between people and plants through the carbon cycle. So cities and other human activities have emitted a lot of carbon dioxide. We all know that. But what some people don't know is that the amount of carbon dioxide in the atmosphere is actually a lot less than what we've put there. And a big part of the reason why is because of forests like the Thain Family Forest at the New York Botanical Garden. This forest and other forests around the planet have basically soaked up about a quarter of all the carbon pollution that we have pumped out into the planet. So this is a really phenomenal realization. Forests are strong partners in the fight against climate change. So my goal at the New York Botanical Garden is to use our incredible collections, the brilliant people, and the strong partnerships to try to solve environmental problems with plants and for plants. So with plants means leveraging the power of forests like this one to store more carbon, and to make communities more resilient. For Plants basically focuses on rare species like the ones that first got me interested in research botany and trying to make the planet a better place for those species in addition to our own. So how do I do that? I kind of have a bunch of different research areas. I'm a bit of a jack of all trades when it comes to botany. I'm interested in urban forests. I'll talk about that today. The details of carbon sequestration. I work in mangroves, which are really powerful ecosystems to protect us from the effects of climate change. And I do work in conservation biology. So let's focus a little bit on urban forests. I'm going to show you an example that you might not recognize. I mean, those of you who are botanists almost certainly do. This tiny little tree is a tiny eastern red cedar. And if you're not familiar with where I took that picture, that's from the train platform in New Rochelle, New York. That's kind of an intermediate station for me on my way home to Connecticut. And this tree just amazes me. And it brings to mind a question that really motivates my work on urban forests. And that's, are cities, trees included, tough and smart enough to adapt to climate change? This tree is absolutely smart, or excuse me, absolutely tough. It's hard to imagine a tougher tree than one that's growing literally under the railroad tracks. Is it smart for an eastern red cedar to grow in that particular kind of habitat? Or is it smart for the MTA to let those plants grow there? Maybe a little bit more debatable. There's clearly some work to do here, but fortunately, the work is being done by brilliant people at the New York Botanical Garden and all over the place. So the real objective that I have in this work is to build on the incredible resources that we have at NYBG, specifically the Thain Family Forest, to have it recognized for what I believe it is. That's the world's premier urban research forest, just by building up partnerships at the garden and beyond. And the pictures that I have here are some of the great colleagues that I work with at NYBG, from John Zeiger, who recently joined us as the manager of the Thain Family Forest, Todd Forrest, who's the VP for horticulture, Melissa Finley, who is the curator of woody plants, who keeps our collections healthy, and then my boss, Eric Sanderson, who's an expert in the historical ecology of New York City. So this is part of our brain trust, part of the people who are working so hard at NYBG to protect our collections. And these are also some of the organizations that have been amazing to partner with in the work at the Thain Family Forest and in urban forest ecology more generally. So I'm going to get a little bit more specific about the Thain Family Forest and NYBG and some of the great collections that we have. So as I'm sure all of you know, we're a 250-acre National Historic Landmark that is really right in the heart of Bronx Park in the greenest borough in New York City. And right in the heart of the Botanical Garden is the Thain Family Forest. This is the largest and arguably the only extensive old growth forest remaining in New York City. And there's really a chance that it's one of the very few old growth forests anywhere in the northeastern states. It's pretty extensive. It's about 50 acres or 20 hectares if you're into the metric thing. And this particular forest has some kind of odd soils that I'll talk about in a little bit more detail in a moment. It's old growth. That means it's never been cut. It's never been plowed. That might make it seem absolutely pristine, but it's also an extremely urban forest. There are about 1.5 miles of active trails, and we get over 1 million visitors to the Thain Family Forest every single year. So this is both a very urban space and a very natural space. And I think that's part of what makes it so dynamic and exciting. So you might think with all this urbanization that this place has seen a lot of change, and that is absolutely true. So since the foundation of Bronx Park in the late 19th century, there have been a host of extreme weather events that have impacted New York City, the garden, and the forest specifically. It just boggles my mind. Almost 30 hurricanes have hit the Thain Family Forest in this period of time, along with a host of other extreme weather events. So folks in Florida are depending on resilient forests right now to protect them. The Thain Family Forest has been that forest in the New York Botanical Garden for millennia, really, but specifically since the garden's been protecting it over the course of the last 120 years. But it's not just extreme weather, which has challenged the plants and the people in the forest and their relationships. There have really been a parade of pests and pathogens that have affected trees, both large and small, in the Thain Family Forest, as they have trees and forests across eastern North America, from chestnut blight, which was first collected by NYBG scientists after it showed up in the Bronx Zoo, to beech leaf disease, which has explosively spread through eastern states. All of these important tree diseases affect individual trees that are in the Thain Family Forest. So between the effects of urbanization, extreme weather, and introduced pests and pathogens, in many ways, the Thain Family Forest is a microcosm for the kinds of impacts that influence forest dynamics in any city around the world. And I want to put a finer point on that just by talking a little bit about the history of the place. So this goes all the way back before the New York Botanical Garden to the 18th century, when the Lurland family purchased this chunk of land to establish their snuff mill. 100 years later, Bronx Park was established, and the founders of the New York Botanical Garden specifically chose this site because the Thain Family Forest was so unique and so scenic. So the research in the Thain Family Forest basically started right with the establishment of the garden, and our first flora was published in the late 19th century, before any flora was published for research forest anywhere in the United States. The research work in the Thain Family Forest got a kind of shot in the arm during the Great Depression, believe it or not, when New York employed people to census every single tree in parks across New York City, including in Bronx Park. So this gives us a really early and very detailed picture of what the forest looked like almost 100 years ago. Later, researchers at the New York Botanical Garden in the Institute for Ecosystem Studies started examining the forest in a little bit more holistic sense by looking not just at the trees and plants, but also those organisms' interactions with the soil and the atmosphere. So they did some really phenomenal work that I'll allude to a little bit later on. So after the Institute for Ecosystem Studies moved up to Millbrook, the New York Botanical Garden wound up invigorating its research program at the Thain Family Forest through the work of really our horticulture department, which started a continuous forest inventory program that's ongoing, where a subset of all of the forest is measured every 5 years, and through some other really great programs like the Education Department's Community Science Phenology Monitoring Program, which is one of the first such research projects really anywhere in the United States. And most recently, in 2008, donors supported the Thain Family Forest Program, which has supported a lot of the activities that I'll be presenting today. So that's a bit about the history. And what does the place look like? Well, it's changed a lot. And you can see from these sets of maps representing dominant species from the first inventory of trees in the Thain Family Forest in 1937 to the follow-up inventory that was conducted by IES scientists in 1985. We've seen a big decline in hemlock that interestingly predates the spread of hemlock woody adelgid, as well as the expansion of some other species that basically filled the gaps left by those once dominant trees that started to suffer some declines. So this is an example of some of the information we can get at the Thain Family Forest, thanks to the incredible history of research that's been done there over the past 125 years. So let's get a different perspective on how the forest has changed over that period of time. So 100 years ago, the forest was still a forest, but the Bronx really didn't look the same. And obviously, the Bronx looks really different now. Really, the Bronx Park and New York Botanical Garden, the Thain Family Forest, are kind of a green oasis in a very urbanized landscape. So you can see this on the ground the next time you visit the New York Botanical Garden. And because we have so much research and so much documentation, some of the comparisons are really stark. So what you can see here are two pictures taken more than 100 years apart from almost the same exact vantage. If you look really closely here, you can see there's a big rock outcrop on either side. What you can't see are really any trees that are the same in these two pictures taken 100 years apart. So clearly there's been a lot of change in the forest, and you've heard about a lot of the drivers of change: urbanization, extreme weather, pests and pathogens. Now I want to see how you think the forest has actually been changing. And we're going to test how your assumptions, or maybe what you know about this forest, compare to the research that has been very recently published using this great body of work. So this is kind of a fun throwback to my professor days. If you don't mind indulging me, you can go to gosocrative.com, or you can scan the QR code here. It will take you to a little quiz that will hopefully allow us to do a little bit of interactive work to set up the next part of the talk. Cool. So I can see a couple of people have already registered. I'll give you all a second to see if you can log on before I drop the screen. And those of you who have logged on will see the first question. We'll go through two of them, and maybe we'll get to the final one as a little bit of a bonus after the talk if there's too much time and not enough questions from you all. Okay, great. So I can see that most people have managed to log on, at least I assume some of the folks who want to do it. So here's the first question for you. Among all the trees that are greater than an inch in diameter at breast height in the Thain Family Forest, stem diameter has blank and density has blank since 1985. Okay, so what I want you to think about is, have trees gotten bigger or smaller? And has their density either increased more stems, or has their density decreased fewer stems since the really thorough inventory that was done about 40 years ago by IES? So I'm getting some solid results from the folks who decided to play my little game here. And most people who answered the question expect that the diameter of trees in the Thain Family Forest has decreased since 1985, and the stem density has increased since 1985. So what do we see? Well, people had a pretty good well, actually, what I should say here is that people had things kind of flipped. Okay, so there really has been no significant change in either one of these metrics in the very recent past, right? Stem diameter has dropped a little bit since 1985, and stem density has dropped a little bit too. So I think I maybe garbled that a little bit. Most folks were right on. So the diameter has decreased a little bit, and stem density has increased a little bit. But that's not to say that you can't still find some absolutely gargantuan trees in the Thain Family Forest, including this spectacular northern red oak, which I just recently measured at about as wide as I am tall. Okay, so there have been some drops in the average size of trees and some increases in their density, but those changes are probably a lot less dramatic than what you might think. And those are only relevant for some of the smallest plants. Whereas if you look at the really big ones, actually the ones that you can see over here, the forest has stayed pretty much the same as it has been since people have been looking at it since 1937. Okay, second question for you. We've talked a little bit about the structure, and the structure of the big plants has been very resilient, even if the little plants have shown a little bit more change. The second question for y'all here is, since 1985, have Thain Family Forest understory richness, so that is the number of different species, has it either increased or decreased? And the proportion of native species, has that increased or decreased? Okay, so now we're on to the next question. Okay, so basically, do we have more species of plants or fewer species of plants? And has the proportion of those species which are introduced either increased or decreased since 1985? All right, so most folks who are, again, playing my game have had a chance to register this one, and they definitely, there's almost a consensus here. I think, with the exception of one or two people, folks think that species richness has gone up in the Thain Family Forest, and they think that the proportion of native species has decreased in the Thain Family Forest. And if I think through a plausible explanation for those two sets of predictions, it's that we've added more invasive species, right? That increases richness and it decreases the proportion which are native. What we've actually observed over the course of the past 40 years or so is that species richness, as people predicted, has increased, while simultaneously the proportion of plants in the understory which are native has increased. And a big part of the reason for this shift, which is probably a little different from what most of you were imagining, is that we actively manage the forest. It's an urban forest. It's part of the New York Botanical Garden. And the Thain Family Forest program has some goals. One of the goals is to increase the proportion of native plant species. And so we see that native plant species richness has increased and the proportion of native plant species has gone up. So whether you're looking at the resilience of this forest to these incredible threats from climate change and introduced species, or you're looking at how well this forest might represent the native flora of the New York City region, there are a lot of surprises. Really, after 125 years, we still learn from this forest. And what I'm going to spend the rest of my talk explaining is a little bit about what makes this forest work. Really, it's people and soils. And so that's what I'll focus on for the rest of my talk. I'll really be focusing on a project where we're starting to leverage this incredible information that we have from above ground to see how it's paralleled by variation that occurs below ground, including some of the incredible opportunities that we have in the Thain Family Forest to look at changes through time based on this record of research that's been going on there for 40 to 125 years. So the specific project that I'll talk about has three different objectives. The first one, as I just explained, is to kind of test how well changes in the plant community reflect changes or variation in soils beneath them. Second objective is to see how these changes in plants and soils relate to the hydrology of the Thain Family Forest, basically how water is moving through it. And finally, to think about how long-term changes in soil health relate to the health of the community of people in the Bronx. So let's tackle the first objective. It's the one we've made the most progress on so far. And a lot of the progress that's been promoting this is thanks to a grant from the Forest Ecosystem Monitoring Cooperative, which is a program that's supported by the USDA Forest Service at the University of Vermont. So the preliminary results that I'll be sharing are part of a grant that's called Enhanced Ecosystem Monitoring in New York City's only old growth forest. Okay, so let's talk about these relationships between what's happening above ground in the forest and what's happening below ground, specifically focused on carbon. The way that we approached this was to try to line up our sampling strategy with what's basically a global standard. The standard is established by the Smithsonian Institution Forest Global Earth Observatory, that's Forest GL, and they maintain a network of large forest plots all around the world, really concentrated in more natural areas, famously at Barrow, Colorado Island, which is in the Panama Canal. So research there has generated tons of insights about forest dynamics, and we're hoping to join that network by following their protocols as carefully as we can. So we use their system to spread a set of 20 by 20 meter plots, about one every hectare, so that we could capture every different soil type that we know is present in the Thain Family Forest. We set our plots up also so that we could maximize the overlap between our soil sampling sites and the vegetation sampling sites, where researchers at the garden have been really focused on changes in plant communities, especially over the course of the past 20 years. And in each one of these sites, we're measuring a suite of features of the forest floor, starting with the litter. So grab all the litter, measure its depth, weigh it. Once the litter's peeled off, we look to see if earthworms have been active based on the presence of their castings. So once the litter's off the soil and we've had a chance to check for earthworms, just as a sneak peek or a preview, earthworms are absolutely everywhere in the Thain Family Forest. There were only about 3 sites out of maybe 197 where we didn't actually find castings. Okay, so we got the earthworms. What do we get to next? We take a soil sample and we get a couple of different kinds of information from the soil sample, as you can see from this gouge auger that I'm holding. This gouge auger shows you some of the stratification of the soil with a very dark brown organic horizon at the top, as well as some changes in the mineral soil that's underneath that organic horizon. Now, not every core is this stratified. A lot of them show a lot of mixing. It's one of the interesting results that we'll be trying to sift through along with the soils that we collected. But the basic thing that we do here is we split our soil core into a couple of different vertical strata, because a lot of what's happening in the soil depends on where you are in relation to the surface. So this approach to sampling the soil gives us a lot of samples and a lot of information. And then we need a little bit more to actually know what's going on with the carbon concentrations. I love this picture. Big shout out to Marlin Coe. We've got a really talented team of photographers at the New York Botanical Garden, and they make soil sampling look way cooler than it is most days. But here I am with a soil bulk density auger, and this is a way of getting a fixed soil sample so we know precisely what its volume is and can then process that soil sample to learn a little bit more about what's happening in the soil after the fact. So we can take these two different kinds of samples with our gouge augers and our soil bulk density augers to get complementary information about what's making up the soil and what its physical characteristics look like. So I was trying this before with Catherine, and when I tried with Catherine before, this amazing video made by our amazing intern Fiona Chow showed up, and she does an excellent overview of our laboratory methods. But when I tried it a second ago, I couldn't get the mic to work, I think because Zoom recognized that it would probably cause ear-splitting feedback to everybody. So I'll just skip it and point out that I had an amazing team of interns, Fiona included, who is a rising senior at the Taft School, who were helping not just collect the soil samples, but also process those soil samples in the lab. So here's kind of an overview of what you see as you start to process one of these soil samples. We pull it out, we press it through a sieve, the soil winds up in the bottom. We process that soil in a couple of different ways depending on what information we want to get about it. In these trays, you can see some of the variability that you get that's based not just on color, but also on texture. These soils are actually pretty beautiful when you pull them out and you look at them closely. But it's not just soil that's below ground. The material above the sieve here falls into a couple of different categories. One of those categories is roots, and those roots are things that Fiona and the team did an incredibly meticulous job of sorting out into a couple of different size categories. So you get a sense for below ground what all these plants are doing with respect to the quantity of roots that are in the soil. And we also sieve out a couple of other things that occur below ground. As you might expect, in New York City or really anywhere, there are a lot of rocks. Some of these places more rocks than others. And this bucket is full of all the rocks that we found in all 197 samples that we had to work through. This bucket that you can see on the top is a little bit of everything else. This includes mulch, it includes fruits that get incorporated into the soil surface. It's an urban site. There's actually a decent amount of glass that we found too. So our soils, our below ground samples, are really a composite, an interesting composite. It includes soil per se, roots, rocks, and other kinds of material. And by looking at the proportion of all these samples, we can really start to get a sense for what's going on below ground and how those below ground features relate to the above ground dynamics that really are what I'm passionate about and what our visitors at the garden are passionate about. So I want to share a few preliminary results, and these are super preliminary, so please do take them with a grain of salt. We'll talk a little bit about what's actually below ground, so the relative proportion of those different features that I pointed out before. We'll look a little bit into how far those roots go. What's their distribution vertically in the soil profile of the Thain Family Forest? And what do we think those roots are going for, right? They are important organs in plants that acquire absolutely essential resources. So can we explain what kinds of resources they're looking at based on their distribution in the soil? Okay, so let's start with what's actually below ground. This ternary plot shows, well, kind of an oh duh answer. Most of what's below ground is soil. So what you can see here is the proportion of these three different below ground components: soil, stones, and roots. And the vast majority of these plots are concentrated by 100% soil, with some of the variability being contributed by stones. That's the next most important fraction that we're dealing with in the Thain Family Forest. And really almost none of it is roots. Even though almost none of it is roots, as you might expect, being a plant-focused audience, the roots are pretty important. So how far do those roots go down beneath the soil surface? Well, we can look at the ratio of roots to soil and see that ratio decreases predictably with the depth of our samples. The vast majority of our roots are concentrated in really the top 2 feet of the soil when we can even get that deep. So you've probably seen these imaginary images like the one that I pulled off of the web in the inset picture that sort of imagine a root system as if it's just a tree projected below the soil surface with all the leaves stripped off. Definitely not the case. The trees shoot up above ground and they spread out at the surface below. So the vast majority of what's going on below ground biologically involving plants and their roots is really concentrated in the top 20 centimeters. If there's soil deeper than that, you basically don't find any roots down there. Okay, so we know that the soil is mostly soil. We know that the roots are mostly in the surface of the soil, but can we actually predict where we'll find more roots? And the answer is kind of yes. Samples that had higher moisture when we collected them had a higher root to soil ratio, meaning that the roots were really concentrated where the primary resources that they provide to the trees in a forest. They are growing where water is most available. So these 3 results that we have are not earth-shattering. It's really just some confirmatory evidence that our methodology is generating a picture of the forest below ground that's consistent with what we'd expect and might provide us some insights into how things are changing above ground. But also this picture of the forest gives some insights into some of the other things that are going on in the forest ecosystem that affect its resilience and the resilience of the Bronx more generally. So I want to talk about our second objective just briefly, even though we're only just getting started. The second objective here is to really think about what these trees are doing to water in the soils and think about how water in the soils of this forest and water in soils around New York City are important for people in the city. So trees drink a tremendous amount of water. And collaborators who've been working with us in the Thain Family Forest at the New York Botanical Garden have an incredible perspective on how trees are doing that. So I'm highlighting a great colleague at City College of New York, Dr. Nick Steiner, who's PI of a NASA-funded project that's ongoing at the New York Botanical Garden that's using remotely sensed data from an instrument that's on the International Space Station that at a very fine spatial resolution measures variation in temperature and connecting that variation in temperature around New York City to variation in the tree canopy. And not just variation in the tree canopy, we know where there's more trees, it's more shady and therefore more cool, but variation in how these trees are functioning in terms of how they're soaking up water, water that ultimately comes from their roots. This picture of this red oak that you can see here is really great because it illustrates a fabulous piece of technology that Nick has built for our forest. These are sap flux sensors. So they're monitoring in real time how sap is moving through these trees from the roots to the leaves. And the roots to the leaves, that continuum of transpiration basically is pumping water out of the soils and pumping water out of the surface of the soils, as we know based on our examination of the root profiles. And this is a really important place to measure how trees are using water because it happens to be the only place in New York City where we have a stream gauge. The United States Geological Survey maintains a stream gauge on the Bronx River in the Thain Family Forest, which is the only place where we're measuring any stream in any part of New York City. And this stream gauge gives us a lot of information, not just about the pulses of water through a tree every day, but the pulses of water through the city when we have a major rain event. So I've got a profile here from part of the last year, and you can see a couple of big spikes. The biggest spike here was a day when I couldn't even make it into the New York Botanical Garden because flooding had shut down Metro North, which is my usual way of getting to work. The image that you can see here is one showing how the water had spread out of the Bronx River and across the Bronx Parkway, affecting not just the commutes, but the lives of millions of New Yorkers. So this connection between roots, soils, trees, and water and watersheds is a really vital one. And it can give us insights into something really important that these forests are doing for us. this urban forest has basically kept about 800 statues of liberty's worth of water, because that's like a weird metric, but basically a tremendous amount of water from the Bronx River, because these trees intercept it from the soils before it has a chance to run off and get into our rivers. So this is a really important service that the forests provide and that urban forests provide around New York City, and something that we're excited to dig into a little bit more based on this research that we're currently doing at the Thain Family Forest. So we get floods like this. What happens to the soil in a place like the Thain Family Forest during one of these floods? Well, this really gets to the third objective for this research project, which is to connect long-term changes in soil health to urban public health. And this is an area where we're really leveraging the incredible record of research that's been done in the Thain Family Forest over the course of the last 40 years. So Rick Puyat, who's a former head of the Ecological Society of America and an emeritus researcher at the US Forest Service, did a big chunk of his PhD at the Institute for Ecosystem Studies at the New York Botanical Garden. And working on an urban to rural gradient, established some of the foundational research on how urbanization affects natural habitats, and specifically how it affects soils. So what you can see here from his dissertation is that there is an extremely predictive and consistent relationship between how many cars are surrounding a forest and how much lead winds up in the soil. And what he found at the time was that the concentration of lead in the soils in the Thain Family Forest was almost a quarter of the action limit that the EPA has for any public space. So there was a lot of lead in the soil, and there was a lot of lead in the soil because there were a lot of cars on the street. And in the late 1990s, this reflected the fact that a lot of the tailpipe emissions of those cars included a lot of lead from leaded gasoline. So fortunately, research at the Institute for Ecosystem Studies and other places supported Clean Air Act amendments that eliminated lead from our gas, eliminated it from our tailpipes, and kept it from being dumped into our habitats. Where did that lead go? It must have gone somewhere. And what I suspect is that that lead has eroded out of the Thain Family Forest as it has out of forests all over New York City and into our waterways. In this particular case, that lead is probably stuck behind the Bronx River Dam. And this Bronx River Dam is actually the focus of an interesting restoration effort that has been proposed by the US Army Corps of Engineers. They want to remove the dam, and their plan to deal with the sediment behind the dam is to just let it wash through the Bronx. So to understand how the health of the forest and the health of its soils, and specifically the lead in its soils, relate to the health of the broader community in the Bronx, been very fortunate to enlist the help of Dr. Pearl Eggendorf, who is a professor at Pace University. She's an expert on soils and urban soils specifically, and thinking not just about their chemistry and biogeochemistry, but their public health consequences. She was a longtime member of the compost team in New York City, and she is an incredible project to understand how variation in soil health can affect public health in this particular context. So that was a bit of a large tour, starting with me and what makes me tick, to this incredible collection that we have at the New York Botanical Garden, and a specific research project that we are right in the midst of pushing forward with a couple of objectives, some of them more scientific and some of them very applied. But I hope what you get from all of them is that what really makes our forests work are plants and people. And this particular project has been a joy to work on, thanks to the great people that I get to work with. And I want to name specifically the manager of the Thain Family Forest, John Zeiger, who's co-PA on the grant that we have from the Forest Ecosystem Monitoring Cooperative, and the team of interns that I had worked with at the New York Botanical Garden this summer. Hardworking, excellent collaborators, excellent communicators. And if anybody's going to be hiring somebody, I couldn't recommend these people any more highly. So that's the end of my talk. Thank you so much for your attention, and I am happy to answer any questions that you all might have for me.
Thank you so much. That was so interesting. I was wondering if the video that you weren't able to play is like on NYBG's website somewhere. Like, can we get a link to it and share it?
Thanks for asking, Katrin. I do have a link to it because Fiona has her own YouTube channel. And if you sift
Oh,
through
okay.
all of her videos about getting fives on all of her AP exams and how to crack the code, like you will find this awesome video
Okay.
about the methodology that we taught her to use for sampling soils at the Thain Family Forest. And it's also
Okay.
got a fun interview that she did with me. So I'm
Cool.
happy to
Okay.
share that with anybody who's interested. Fiona is an absolute all-star.
Cool. Okay. All right. So for questions in the chat, Brad, if you don't mind, can you see the chat? Okay.
Yeah,
Okay.
and
Do you mind
hopefully,
just reading them out before you answer so that the like question is recorded for the recording? Yeah.
no
Okay.
problem.
Thank you. Appreciate it.
That's a good question. Thanks, Tom. And Tom's question is, has the Army Corps actually measured the sediment behind the dam and if it will wash down and where it will go to? So, Tom, the short answer is no. They haven't measured those sediments. And from what I understand, it's not part of the uh, fish passage project that they had proposed. And that is not negligence on their part. From what I understand, that's kind of standard practice for dam removal projects in the Northeast. There's been some research, particularly for dams in more rural areas, that full removal and sediment redistribution is one of the healthiest things that you can do as part of the decommissioning of a dam to promote fish passage, which is what the Army's Corps' big objective is here. They want to just let more fish get from Long Island Sound up to the breeding grounds in the Bronx River. But we believe, and I think there's good reason to believe, that it's probably a different story in an urban setting. And the history of pollution probably makes that so. So our hope is to have the data, the resources, and the capacity to partner with the Army Corps to better understand the kinds of risks that might come from this particular approach to implementing a fish passage project. And it's not that I or anybody else at NYBG hates fish. I mean, I kind of love fish. I would love if fish were able to move around in this landscape a little bit more easily. But we have a responsibility for the people who live in the rest of the watershed. And I think there is some chance in this particular context and in other urban watersheds that this dam removal without testing and hydrological monitoring could potentially expose people to harm. And the unfortunate thing about the communities that live along the Bronx River is a lot of those communities have faced a lot of environmental injustice over a really long period of time. And the very last thing we want to do is to exacerbate that. So that is one of our objectives. So excellent question. Thank you for asking. It's one of the things that we're working on, and we're hoping to partner with the Army Corps to do this in a good, scientific, and responsible way. Great. So I can see that Margaret also asked a question. Thank you, Margaret. Margaret's question is, what do you know about the history of earthworms in the forest? Margaret was recently surprised to learn that earthworms are not actually present in previously glaciated areas, which I assume the forest is. So I'll kind of start at the end of your question and work my way back. You're absolutely right. It was glaciated. You can see the potholes. You can see the striation marks. It was very much part of the geologic history of this area, and I'd encourage you to come and check out for yourself. So it was glaciated, and you're also right that earthworms basically weren't present in North America in those soils that had been recently glaciated. So all the earthworms that we have in the Thain Family Forest are introduced earthworms that I don't know enough about. I have no experience with earthworm taxonomy, and we had chunks of earthworms that showed up as sort of gross, very much Halloweeny, in all of our soil samples. Not quite all of them, but quite a few of them. And so I was interested to see if they were as prevalent in the Thain Family Forest as they are in other forests around Eastern North America, where they've been introduced and where they spread. I was maybe thinking there was a chance that the urbanization had kept them from coming in, or that some aspect of the management had kept them out. but they are pretty ubiquitous. They're so ubiquitous, in fact, that it's really going to be hard to see if they affect variation in soil features because they're kind of everywhere to begin with. So yeah, lots of earthworms. I expect that they do have some impacts, but it's going to be hard to figuring them out just by looking at the Thain Family Forest because they are so ubiquitous. Great. So I can see that Jeremy has a question with his hand raised and that a couple of other folks have asked questions as well. But Jeremy, I invite you to unmute and go ahead and ask your
Yeah,
question.
yeah, I actually have a follow-up to that. You said you weren't sure about this species, but uh Amenthus agrestis and that genera of the called like the snake jumping worms. I was wondering if you had noted them particularly also because they're a problem in Northeast forest, they're spreading, and they actually change the leaf litter composition in forests that they invade. So I was wondering if
Yeah.
you had noted them in there or had plans to.
Thanks, Jeremy. I've heard about those. What a terrifying name. It's really hard to forget. Like snake jumping worms. No thanks. Uh, Unfortunately, like I'm such a bad invertebrate biologist, I haven't managed to figure them out. I mean, one thing that we certainly could do with a much bigger grant would be do some DNA barcoding, and presumably we could pick that up from a few of our soil samples and get a sense for the invertebrate communities and other aspects of the biology of those soil samples. Um, my guess is that somebody who's good at identifying those worms could probably figure out if they're present in the garden or if they're very widespread in the garden. But one thing that I can speak to that I observed, which is the relationship between worm presence and the leaf litter and duff layers. So, in my answer to Margaret's question, I said that earthworms were basically everywhere. Basically, not absolutely. There are just a handful of places where we didn't find them. And in the majority of those places where we didn't find them, there was a very distinct difference in the soil. And the difference in the soil was that there was a big duff layer. So not a lot of leaf litter, but a lot of that kind of partially broken down plant material that sits on top of the soil. It was really only where the earthworms were absent where we found those kinds of soil samples. And I think there are probably some very micro topographical areas in the Thain Family Forest, basically crotches in between big rocks where the worms can't get. And those are the places where we found those kinds of soils and where we didn't find many earthworms. So I think if we did a different sampling project, one that was really focused on the earthworms, I'd bring somebody on board who could figure out what those terrifying snake ones are. And B, we could get a sense for where they're present and where they might be moving around. And I'd like to point out that that's just an example of one of the fantastic research questions that I would encourage anybody, regardless of your background, to engage in at the Thain Family Forest. Like it's a space for researchers to ask great questions because it is so unique. And we're learning all the time from the community of researchers who are working there. We have something like 15 active projects that are going on right now that don't include the ones that I'm working on with other garden staff. So thanks, Jeremy, for that. This is great. I see a few other questions and we've got a little bit more time. So Amy asked if anybody is doing survey work with other soil invertebrates. Not exactly. So my colleague, Dr. Rob Noxie, who I'm sure many of you know as our curator for the North American flora, he is also really into beetles. So Rob has had an ongoing beetle inventory that he's been doing in the Thain Family Forest for a pretty long period of time. I think a lot of those beetles are deadwood associated, even though I know he does a lot of the trapping in the soils based on his pitfall traps that I've looked at. So Rob has a pretty good record for the beetles and how they've been moving around. I also believe that there's a researcher at Fordham University who's been looking at ants in the Thain Family Forest. So we've got those two different insect groups, but I'm not aware of anybody who's been looking at invertebrates more broadly. I will point out though, Rich Puyat, who I give a big shout out to, the former ESA president who did that foundational soils work, a big part of his dissertation research was actually looking at the soil invertebrate community. So we do have some historic soil invertebrate data sets. And if that's something that you're really interested in, I'd be glad to share some of that information with you. So I can see a couple more questions, but I will point out that we're right here at 7. So I'll let Katrin sort of be the judge of whether we keep chatting. I'm glad to do that for a little bit. So what do you think, Katrin? Should we answer a few more questions or?
Yeah, if you have time, maybe we can take this question about the the little known facts or disprove a myth, and then I'll have one final question to wrap us up. Does that sound good?
That sounds awesome.
Okay, all right.
So thanks. And I just want to point out that Helen in the chat, who is a fantastic resource to have in the community. So hi, Helen, thanks for participating in this talk. Is aware of some really cool research that I didn't know about on
What's that?
on urban rural gradient work on worms done by apparently a researcher at Fordham, Margaret Carriello. So thanks for sharing that, Helen. And folks who are into the kind of worm dynamic hopefully can follow that thread a little bit. I sure should. So I can see one other question here from Aver. Aver asked, what is something within my expertise that you wish the public knew more about, like a little known fact or to disprove a myth? Okay, so I mean, it's kind of a question of how much folks know. I think one thing I led off with at the beginning of my talk, the fact that forests do so much good for us in terms of protecting us from the effects of climate change, that is known, but it's kind of disputed. And I'll try not to get too wonky about it. But a lot of it has to do with carbon credits. People are really concerned about how those markets are working. They think that it's a bunch of greenwashing. You know, it's letting companies and other organizations avoid the hard work of actually decarbonizing by basically just saying they're going to plant a bunch of trees. And people think about those trees and they think tree's going to die. It's never going to store any carbon. That line of thinking, I can understand why people have those concerns. And I think there are good reasons to look critically at carbon offsets and carbon offsets associated with forestry and afforestation. But it is so vitally important that we work with nature to solve the climate crisis. And trees have been doing so much for us, and not only for us, but for so many other kinds of organisms. Really figuring out how to manage our forests better is a vital part of the solution to climate change. And that is a fact that I hope everybody takes away from this talk. And I hope you can execute it in your love for plants in your own community. Like, really plant a tree. Like, there's some situations where it's probably a bad idea. So, you know, talk to an arborist or somebody else who has some experience in forestry before you go ahead and do it. But it's a solution that's accessible to most people, and it will do a lot of good. Great.
Thank you so much. That was a lovely, I think, question to end on. And my last question is usually like, what's one thing that you want people to take away from the talk? And I think that was a very lovely answer, and we can end it there. So thank you, Brad, so much for your time. Thank you everyone for being here. Please check out torreybotanical.org to hear about our upcoming events, and our next Torrey talk will be in November. Lydia Paradiso will be talking about some historical botany of the New York City region. So thank you everyone, and have a good evening.
Thanks everyone.
Bye.